Let's go. So welcome to the first workshop on amino acids and proteins. As you know, I'm Dr. Phil Carpenter, and I'll be taking you through this. So today, this is the outline. We're going to look at some content review. I'm going to go over my theory about amino acids. We'll talk about memorization. We'll talk about structure, application, function, chemistry, etc. And so the material that you're going to see today is derived from our amino acid, peptide, and protein diagnostic module. All you guys have access to that. So there's 86 questions. We're going to do about 13 questions today, and what we're going to be doing is under our workshop function. So you've all signed up, you have an account, and so therefore what you're going to have is the ability to go to the workshop function, and we're going to do the hemoglobin structure and function and the DNA polymerase structure and function passages, because they're going to speak to not only amino acids, but the application of them to a variety of scenarios. And so we all know what amino acids are, right? They have an amino group, as you see here, the NH3+, and you have a carboxyl group. Now, as shown here, this is a Fischer projection, and what you see is the alpha carbon designated by the arrow. And in the Fischer projection, the most oxidized carbon is always at the top by definition. And in the case of serine, there's the D and the L isomer. So we won't get too much into the stereochemistry of amino acids today, but we should recognize that humans only use the L version of the amino acid. They have the enzymes that can only look at L. And so the D and L here is designated because if the amino group is on the right, it's D, and if it's on the left, it's going to be the L isomer. And so probably one of the most important things with respect to amino acids is acid-base chemistry. And given the fact that our bodies have to regulate pH to such a narrow window, like around 7.4, if you're above it or below it to any significant extent, you're going to be really sick. And so what we see here is a dissociation for a Bronsted-Lowry acid. So we're going to be talking about amino acids in the context of Bronsted-Lowry acids, but we also realize the MCAT looks at the Lewis acids as well as Arrhenius acids, and we have a bunch of stuff like that in our physical sciences section. So what you see here is an equilibrium expression at the top. We have an equilibrium constant, and if you take the negative log of the equilibrium constant, you get what is defined as a pKa. And the pKa is designed so that people can deal with easier numbers, like on a 0 to 14 scale, as opposed to like 5 times 10 to the minus 10. Those are harder numbers to deal with. You should appreciate that the larger the equilibrium constant, and the larger the K, that what you have is more product lying to the right of the equation at equilibrium. So if you take a look down here at this amino acid on the left, what I've shown you, this is glycine. It's a simple diprotic amino acid. It has two acidic groups. It has the carboxyl and it has the amino. And the carboxyl pKa is about 2. And the amino pKa is about 9. You should remember that, but you should also remember that these are not exact because the numbers change as a function of ionic strength and other physical parameters like the dielectric constant. And so what happens then, as you increase the pH going from left to right, what you do is you titrate out the lowest pKa because that's the most acidic group. And then ultimately what you end up with when you do that is you end up with the isoelectric point. And what you see in the middle here is that this structure has a plus and a minus. Its overall charge is 0, so it's a Zwitter ion. It's Zwitter ionic. And as you add more base to it, what you do is you actually titrate off the amino group. And what you've done is you've increased the anionic character as you move up in the pH. And so what I think you should definitely remember is that if the pH is greater than the pi, you have increased anionic character, more negative charge. And if the pi, the isoelectric point, is greater than the pH, it's more positively charged. And so what you're likely to see on the exam, and you probably see it in class, is the classical titration curve. So I'm showing you alanine. This is a simple diprotic acid. Actually, alanine is a pretty important amino acid. It has a methyl side group here, and it can be readily converted into pyruvic acid. I'll show you that in a minute and why it's significant. But if you take a look at this titration curve and you have a solution of alanine at really low pH, we know that since the pKa's have not been titrated yet, and as you add more and more base, what you do is you get to this inflection point here. And at this point 1, what you are is you have half of the carboxyl groups have been titrated away. And then as you come further along, what you do is you end up titrating all of them off. And so as you move along from point 1, 2 to 3 to 4, what you do is you move from form A to B to C, and ultimately what you get is point 4, where you're going to be completely deprotonated and negatively charged. And so you should definitely examine your understanding of titration curves, because I feel as though they're going to be on the test in some form. So another thing about these acid-base problems relates to what's called the Henderson Hasselbalch equation. And what you have with respect to this is you have the classical equilibrium shown here, but if you algebraically manipulate it, you can get this equation pH equals pKa plus the log of base over acid. And in this case, we're talking about a conjugate base. I've arbitrarily designated as A minus, but you notice with histidine, it's not negatively charged. So it's just nothing more than a denotation. And so common questions are, what is the fraction of histidine residues that are positively charged at pH 7.4 if you're given the pKa of the side group, the imidazole side group is 6.4. And then what you should do is you can either calculate it out the long way, but you should also get super accustomed to actually doing these the fast way. And the fastest way to do this, you guys, is to recognize that the numbers have been placed such that the math becomes easy. And I would argue to you that a lot of the MCAT math will be of that variety, because you don't have a calculator, so you're going to do things by hand. And so what you'd see then is that if you're at pH 7.4, but the pKa is 6.4, since you've gone past the pKa, you've titrated off these protons to a significant extent. And so we know that whatever the answer is, that the majority of the residues are going to be deprotonated, okay? So what you can do then is you can actually go through the equation. I won't go through all the steps here, but you can actually plug it in. And when you do that, you end up solving for 10 equals the base over the acid. And what you know basically is that 90% of the histidine is in the conjugate base. And the reason why you know that is because since you're 1 pKa unit away from the pH and it's a log scale, that means it's going to be a factor of 10 or 10 to 1, which is approximately 90% of the histidine is in the form of the conjugate base, which means the answer is that 10% is positively charged. You should definitely be practicing these if you're not comfortable with them. And you're probably going to see more complex ones on the MCAT with respect to peptides that'll have multiple ionizable functional groups. So the Henderson Hasselbalch is absolutely a staple of your knowledge. You must memorize that equation or be prepared to derive it. So I'm going to go through a survey of amino acids right now. And Amrit, yes, you could take screenshots, of course. I appreciate you asking that. So let's understand the chemistry of the side chains. Okay, so we're not going to talk about memorizing how to draw out each individual carbon and nitrogen, et cetera, in amino acids, but you should understand the classes and what they do. So here we have acidic, which is aspartate and glutamate. They have carboxyl side chains. They have 2 carboxyls, okay? So the difference is 1 methylene group. And what we also have then is the basic residues are histidine, arginine, as well as lysine. And so it's important to recognize that lysine is a primary amine. And so it has all the functional chemical properties of primary amines. I'm going to show you something with that in action in a moment. So then we move over to the sulfur-containing amino acids. We have cysteine as well as methionine. The double AMC content outline specifically singles out and mentions cysteine. So we have to think about what cysteine can do, and what it can do is going to be a function of the sulfhydryl group, okay? It's a thiol. So it's like an alcohol with a sulfur group, and so we're going to see some similar properties to alcohol and a lot of dissimilar ones as well. Now there's methionine right here, and methionine, as we know, starts the vast majority of polypeptide chains. But I'll show you something about methionine in a moment that relates to vitamin B12. Here's the aliphatic ones, leucine, isoleucine, valine, as well as alanine. And these are hydrophobic. So they have a lot of CH2s and CH3s. And so with that comes the idea of the hydrophobic effect. And so we would expect these residues to lie in the interior of proteins because they want to be away from water because they're hydrophobic. And that has to do with the thermodynamics of entropy, as we shall talk about in the future. So, and then there's glycine over here, and glycine basically is a primary amine, but it's achiral. It doesn't have a chiral center, has no optical activity because this carbon is bound to 2 hydrogens. We're going to move over to the polar uncharged. We see serine and threonine. These are commonly phosphorylated. Serine and threonine are aliphatic alcohols. Their pKa is about 13. So if you're at pH 13, about half of serine would be deprotonated. Over here is asparagine and glutamine. These are amide side chains, okay? And then we move over to the aromatic ones. This is tryptophan, tyrosine, and phenylalanine. And tyrosine is an aromatic alcohol. Its pKa is 10, and that raises the question, why is it lower than the aliphatic alcohol serine? And the reason why is because if you lose this proton, then you have a negative charged oxygen, and those electrons can be resonance stabilized by the ring. And so it's happier through resonance, and that's why it's easier to remove the proton. And then we have proline, which is not even an amino acid, it's an amino acid, so it's a secondary amine. And by virtue of the fact that it's in a circular structure, proline has a lot of pretty odd properties, and we'll talk about some of those. Now, with respect to cysteine, there's a couple things that are going on. If you look in the middle panel here, we see the reduced sulfhydryl groups can be oxidized to disulfide bridges, which provide structural stability to a number of proteins. And the vast majority of proteins that have disulfide bridges, you guys, are actually secreted. They're either on the membrane poking outside to the outside of the cell, or they're secreted, like the hormone insulin is loaded up with disulfide bridges. Now, on the top panel, what you see is that the sulfhydryl group can be ionized. So it has a pKa around 9 or 10, depending upon its molecular environment. And this sulfhydryl, this anion here, is a strong nucleophile. And what it can do is it can couple to a number of things. And in this case, I've shown you the reaction with farnesyl pyrophosphate. What does that do? What it does, it actually attaches, this hydrophobic group, this tail. And when you do that, that tail is now endowed with the property it's inserting into the membrane. And so it's a way to get proteins stuck in the membrane. And the classical one is known as the RAS oncogene. It only works when it's prenylated on a cysteine residue in the membrane. Now at the bottom, I have a little caveat to the idea that there's 20 amino acids. So if you ask somebody, how many amino acids are there? People are going to say there's 20 that can go into a protein, but the answer is actually 21 because there's this crazy thing called selenocysteine. And so what happens is that if you look at the periodic table of the elements, below sulfur is selenium, and you can actually incorporate this into polypeptide chains. Should you know about pyrrolysine as well? We're not going to talk about pyrrolysine, but it wouldn't hurt to know it. And yes, the PowerPoints will be available, you guys. Essential versus non-essential amino acids. Okay. So everyone talks about an essential versus a non-essential amino acid. I'm going to make an argument to you, but it's not as clear cut as that. So let's take methionine, for example. So methionine is made in your body. You take homocysteine, and methionine synthetase, or methionine synthase, turns it into methionine in a reaction that requires vitamin B12. Now, what you'll notice about B12 is that you see it's methyl B12 up here. And then in the reaction, it turns into B12. So now we know something about what B12 does. It provides carbon methyl groups to substrates, and you make methionine. But there's a disease called methionine synthetase deficiency. And if you have that problem, the enzyme doesn't work, then homocysteine builds up. That's potentially dangerous. That means that you're conditionally essential as methionine. So if you can't make methionine, it's a conditional essential amino acid. And in some cases, like a lot of growth from like little babies and stuff growing fast, they don't make enough methionine. So then they have to supplement as well. The second one to look at is tyrosine. Tyrosine is made from phenylalanine. What you do is you take that ring and you oxidize it right there and you put OH onto it. And then tyrosine is a precursor for a number of molecules. One of them is called L-DOPA, and you guys have all heard of DOPA and dopamine. And the class of molecule this is called is a catecholamine. I think this is an important term for you to know because docs know it. They use it all the time. In fact, on the Quest diagnostic form, it says catecholamines. You can measure them in people's urine, and you do it all the time. Now, what a catecholamine is, it's a benzene ring, as you see here, okay, with two hydroxyls next to each other and an amino group somewhere else on the molecule, okay? So that means phenylalanine is a precursor to tyrosine as well as the dopamine, but there's a disorder known as PKU, or phenylketonuria, where this hydroxylase enzyme is deficient. So there's people born that are homozygous recessive for this allele. And what happens then is that you build up phenylalanine, and I'll show you in a minute that it's converted into phenylpyruvate. Okay, phenylpyruvate is actually a neurotoxin when it accumulates too much. So if you have PKU, you can no longer make tyrosine. So then tyrosine is conditionally essential as well in PKU. So amino acids, as the word amino means, is nitrogen. And so what about nitrogen balance? So let's take the example of phenylalanine being converted into phenylpyruvate. This is a pretty high yield reaction. It has a lot of clinical utility. It's something you should know. Let's take a look at it. So we have phenylalanine over here on the left. Okay, we have an amino group. And what this enzyme transaminases do, they use vitamin B6 or pyridoxal. Okay, so it's one of the vitamins on the side of your cereal box. And when you transaminate, what you do is you give up your amino group to a carrier, which is alpha ketoglutarate, and then you become the alpha keto group. So you swap the amino, and then you put the keto over here. So what does transamination do? It removes the amino from the substrate, gives it to a carrier. In this case, alpha ketoglutarate becomes glutamate, and then you're left with phenylpyruvate. So what happens to glutamate? Well, glutamate is going to go to the urea cycle, which I'll show you in a moment. And so we can think of glutamate as not just an amino acid, it's also a neurotransmitter, but it's also the carrier of an excess amino groups. Now, these transaminases are really important, you guys. There's one called ALT for alanine transaminase, and that's actually it's a lot of it's in the liver, not exclusively, but its biased location is in the liver. And so what docs do is they measure ALT for liver function. So it's a liver function test. ALT is one of the enzymes. And what does ALT do? It takes alanine, it gives up its amino to alpha ketoglutarate, becomes glutamate, and then the alanine turns into the cognate alpha keto acid. And look what you have, abracadabra, you've made pyruvic acid, which we know is a very important player in metabolism because you can oxidize the carbons in glycolysis, or you can reduce them to actually make sugar by gluconeogenesis. So ALT and transaminase is super important. So we've transaminated an amino acid, okay? We made glutamate. What happens to the glutamate? Well, you shouldn't memorize this cycle at all, but you should appreciate a couple critical factors. That is, the glutamate will donate its amino group to make carbamoyl phosphate, and then you regenerate alpha ketoglutarate. And that amino group is going to fly through the cycle here, and ultimately what you see is that it's going to make urea. And urea is measured by docs as blood urea nitrogen, and it's only made in the liver. And so urea is measured for liver function as well as kidney function, because the rate at which you excrete it in your kidneys is important to know. So blood urea nitrogen is a function of excess nitrogen that was transaminated and loaded onto glutamate. The other thing about the urea cycle, besides the fact it's only performed in the liver, is that you actually make arginine. You make arginine. We know arginine is in proteins. We know arginine is a nitric oxide precursor. It provides the material for that. So if you have a deficiency in the urea cycle, there's a number of inborn errors in metabolism, then arginine becomes an essential amino acid. One of the things we're going to look at in the passage today is the use of lysine in affinity columns. And so now we're going to speak to actual chemistry. So what guys actually do to make an affinity column, and the next time we get together, we're going to elaborate even further on column chromatography and lab techniques. This is just a slight introduction to it now. So the idea then is to take a bead, and this bead has been rigged up such that it has this activated resin that has an ester group on it. You should recognize that as an ester, okay? And if I took an immunoglobulin, or for that matter, any protein that has lysine side chains, okay, and in the deprotonated state here, you can imagine the pH is fairly high, maybe 10. What would happen then is that these lone pairs on the nitrogen are nucleophilic, and they're going to attack this electrophilic carbon in the ester. And what you're going to do is you're going to covalently link this protein, in this case an immunoglobulin, onto the column. And so what you have then is a protein stuck on a column, and you can pass extracts or things that you wish to interact with it onto the column, and you made an affinity column. And the reaction then is that an ester plus an amine is an amide plus an alcohol. But in this case, our amine was lysine, okay? So what you have to recognize is the chemistry of the side chains and how to use them. So we're going to see this in the passage a little later on. So I'll mention a few more things, and then we'll move on to hemoglobin, and then we'll go to our passages. So the branched chain amino acids, BCAA, are also known as the aliphatic ones. You may have seen them at GNC or on the internet. So what bodybuilders do is they eat a lot of BCAA, or they put it in there as a supplement. And the reason why is because our muscles are significantly biased for having branched chain amino acids as structural components of the tissue. Another thing about amino acids is that they're not just for proteins, you guys, they're actually precursors to incredibly important molecules. And what you see here is the indole side group of tryptophan. And as you follow tryptophan's metabolism into serotonin, what you can see is the indole side chain. So I would argue that you don't need to memorize the structure of tryptophan, but you should recognize that serotonin is derived from tryptophan by the presence of the indole side chain. And so if serotonin, as we know, is a neurotransmitter, and so what docs do is they measure 5-hydroxytryptophan in your blood or urine in order to see if you're defective in serotonin levels. Another thing is the histidine side chain. This is imidazole. If you decarboxylate histidine, you get histamine. And we all know what histamine is because so many of us take antihistamines. Histamine is a vasodilator. It's involved in allergy as well. So with respect to the aromatic amino acids, since they have a lot of pi electrons, what they can do is absorb ultraviolet light. And here you see a UV spectrum of them. So now we're kind of using amino acids and thinking about spectroscopy. So we're not memorizing the structure so much. We're appreciating that the aromatic amino acids have a lot of pi electrons, which are absorbed, they can absorb UV light. You can actually detect proteins based upon their tryptophan and tyrosine content at 280 nanometers in a spectrophotometer. And what that sets up is an MCAT favorite known as the Beer-Lambert Law. And that describes a linear relationship between the concentration of a protein with tyrosines, for example, and absorbance. So if you look over here, if you stick a sample in what's called a cuvette of your protein, and you put light through it, you have an incident light here. And then when you shine it through, as it passes through the cuvette, what happens is that those tyrosines at 280, as well as those other aromatic residues, can absorb that. And then the incident light coming out is going to be lower, less intensity. And what you can define is that the absorbance equals the molar extinction coefficient, which is a property of a particular type of molecular group, or a protein versus like hemoglobin would have a different molar extinction coefficient than serum albumin. And you can determine the concentration using this formula, and the path length, the length at which is crossed, is usually set at 1 centimeters. And so by using the Beer-Lambert law, you can determine the concentration of species in solution with ultraviolet light, and that's a function of amino acid side chains. We've all seen that peptide bonds are in proteins. We know that they have resonance hybridization here, and so they have a planar structure, so you can't rotate around them very well at all. And most of the time they're in the trans configuration, not all the time. I think we've all seen that before. And so the primary structure is determining the tertiary folding structure. And I want to tell you about a really quick experiment we see on the MCAT before. It's called the Anfinsen experiment. He won the Nobel Prize for this, and you're thinking, wow, that must have been an easy Nobel Prize. This is pretty hard to do and revolutionary in the 70s. So we took ribonuclease A, and in its folded state, it has these disulfide bridges you see in the blue. So these are oxidized cysteines that have come together. And what he did was he reduced them, like with dithiothreatol, and then he denatured it. So here it is in the green, it's denatured, okay? And then what he did was he did one of two things. He took half of this material that's denatured and he removed the urea and then he oxidized it again. And what turned out remarkably is that the cysteine residues found themselves and re-oxidized to form disulfide bridges that mimic the native state, okay? But if he did the reciprocal where he oxidized them first and then removed the urea, the cysteine residues couldn't find themselves. They actually made these things called scrambled structures, and they're inactive. And so what Anfinsen proved in this experiment was that the primary protein structure determined the three-dimensional structure of the actual protein, which we know is a tertiary structure. But to get to the tertiary structure, you have local folding regions known as secondary structure, with alpha helices and beta sheets being the most commonly described. And if you take a look at the alpha helix, what you recognize is that it's the actual amide nitrogen that hydrogen bonds with the carbonyl oxygen. You're not seeing side groups actually hydrogen bonding with each other. And remember, what's a hydrogen bond? Well, it's going to be an electronegative atom like oxygen or nitrogen, hydrogen, and another electronegative atom here. One of them is a donor, the one that's covalently linked, and the other one's an acceptor that's hydrogen bonded. And here you see beta sheets as well perform the same type of structural relationship with the hydrogen bonding. But you notice that proline, for example, based on its weird secondary amine structure, it doesn't have an amide hydrogen bond or amide hydrogen to form a hydrogen bond. It'll never be found in these structures. So you should definitely know that proline is called a helix breaker. But that's the reason why. Okay. And so now we'll go to hemoglobin. This will be the last thing we talk about before we move on. So I think everyone's seen hemoglobin. And so hemoglobin is the classical description of the tertiary structure, which is the folding of the polypeptide, and the quaternary structure is when multiple subunits come together to interact. All hemoglobins are tetramers. If you're not a tetramer, you can't work as a hemoglobin. Now this one's called hemoglobin A for adult. So all of us have hemoglobin A. It's alpha 2, beta 2, okay? Now, 95% of a red blood cell's mass is hemoglobin. That tells you what the red blood cell's thinking about. Now, intrinsic to hemoglobin is these 4 parole rings, which have their nitrogens coordinating iron 2, ferrous, not ferric. If ferric is there, it won't work as well, as I'll show you in a moment. So why is hemoglobin so important? Well, it's the definitive case on structure function. It's got the historical relevance, but it is incredibly clinically relevant with respect to anemias, and we'll see sickle cell today. So the med students are always looking at hematocrit, hemoglobin, and other things that are functions of red blood cell. Okay, so hemoglobin is totally high yield. And so hemoglobin exists what's called a cooperative binding curve. This is a sigmoidal curve, okay? And so what cooperativity means is that each subunit interacts with another subunit. And so the 4 of them together form a functional unit. And what you can see here is that to get the first oxygen onto hemoglobin is a little more difficult. And then as you start getting more higher oxygen concentrations, what you do is it's easier to get on. That's a cooperative curve. It's sigmoidal. Now, there's also hemoglobin F, which means fetal. This is what was in our bodies when we were in utero in our mothers. And so what this is the gamma subunit, okay? Now, the gamma subunit is different than the beta subunit, and we'll elaborate on some of that in a moment, but what you should recognize is that gamma is a good substitute for beta. And if you take a look at this, the hemoglobin F tetramer is also cooperative, but it shifted to the left. And if you even include myoglobin, which is a monomer, not a tetramer, that's in our muscles, what you see is even further shifted to the left, but also it doesn't have a sigmoidal cooperative curve. Why? Well, it's a monomer. There's no subunit interactions, but also what it has is your classical hyperbolic saturation curve. And if you look at these curves further, and you should definitely understand how to read these curves, what you'll recognize is that the more left you are, the higher affinity you are. Let's take a look why. If you're at 30, okay, on this partial pressure of oxygen and torus, and you look at at 30, what you see is the saturation of hemoglobin A is about 10% when you come over here, but hemoglobin F at 30 is closer to 40, and myoglobin is almost at 100. And so what that suggests is the further left you are, the higher affinity you have for oxygen. That's the way to think about these curves. Now, let's take a look at hemoglobin and oxygen. And I'm going to argue to you guys, this is an acid-based chemistry problem. And we're also going to see that behind the curtain is going to be an amino acid problem. And so let's take a look. So we're in the lungs now, we're breathing in, and you have hemoglobin binds to oxygen. And I'm going to argue to you that the binding of hemoglobin to oxygen is like an acid. You release a proton. We're going to find out what amino acid is going to release that proton in a minute. So I think of it as like an acid. That's the only way I can put this all together in my head. And once I know that, I can do all the other dominoes, but it took me so long to figure that out. So what we're doing is we release a proton, and that proton then is in solution, and it's actually going to be part of the carbonic acid buffering system, which is the major buffering system to regulate pH in cells. And what happens at the proton binds the bicarbonate, you form carbonic acid, and there's an enzyme called carbonic anhydrase. And what that does is it can dissociate bicarbonate into H2O and CO2. That's the reverse reaction. In the other reaction, the forward reaction, it's CO2 plus water. And remember, enzymes can go in either direction. Okay, so we got that. So what happens is that if you take a look at the hemoglobin tetramer, that when it has zero oxygen bound, it's called T. T is for tight or taut. And it's held together by these salt bridges, these electronic interactions that are hard to break. So in order to get the first oxygen on, if you take a look at this, you have to break this one here as well as that one there. So you have to break two salt bridges to get the first one on. That's why it's the hardest one to get on. And then you start adding them. And then when you ultimately end up with a form known as the relaxed form. And the relaxed form is the form that is bound to 4 oxygen molecules. So then binding of hemoglobin to oxygen releases a proton. Okay. Now, what happens with oxygen is that it's so incredibly reactive that it causes a lot of problems. It causes oxidative damage. So under normal circumstances, the hemoglobin iron is ferrous +2. That's okay. But every once in a while, you can actually oxidize it to ferric +3. That's not good at all because this will not deliver oxygen at all, okay? It's unstable and it won't deliver it. So the cell has evolved a mechanism where the glycolytic NADH is actually used to reduce it back. So people that have Fe+3 have a condition, they have methemoglobin, it's called, okay? I don't know why it's called methemoglobin, to be honest with you, but it's the +3. It does not deliver oxygen at all. So the red blood cell is 95% of its mass is hemoglobin. We know it's involved in oxygen, and part of the problem with playing with oxygen is that you get a lot of oxidative damage. And so the red blood cells also will have a way to fix that as well, but we won't have the time to talk about that today. Okay. You bind hemoglobin in the lungs with oxygen, and then you circulate the peripheral tissue, and imagine we're over by a muscle that's been actively playing basketball or working out or something like that. What is that muscle doing? It's burning glucose, okay? It's going through TCA cycle, it's making carbon dioxide, it's making acids like lactic acid and pyruvic acid, and the CO2 diffuses out. There's no receptor for CO2, It's soluble by Henry's law, so there's solubility in solution, okay? And then what happens is that it combines with water, and now carbonic anhydrase moves the forward reaction to make carbonic acid. That's a weak acid, so it'll dissociate. And when it dissociates, what does it do? It comes over here and binds to oxygenated hemoglobin, and it releases oxygen, okay? So protons cause oxygen to be released. It's called the Bohr effect, okay? So we saw that when hemoglobin bound oxygen in the lungs, it released a proton. Now, when it delivers it, it's taken its proton back, and then it'll circulate to the lungs, and it'll cause the whole cycle to recapitulate itself. So histidine is at the center of the Bohr effect. So down over here, when we bound, we had histidine in a deprotonated state, the conjugate base. It took up a proton, okay, and then it caused it to be released. And so what we see then is that histidine exists between the deprotonated uncharged state, the protonated state, and then when you release the oxygen, you have the plus charge here because you protonated it. And then what happens then is that plus charge can form a salt bridge within the aspartic acid, and now you've remade the T form of it when you got rid of the actual oxygen. And so what it is that histidine is the critical residue for oxygen binding as well as release. So the truth of the matter, you guys, is that it is an amino acid problem in disguise. Now, here's another chart about hemoglobin binding and pH. If you take a look over here, this is the partial pressure of oxygen in a typical tissue over here. If you notice that this is hemoglobin A right here, and then as you move up, you get a higher percent saturation, and as you move to the right, you get lower, or you release the oxygen. That's what you end up doing. So you should take a look at this for sure. So here's the PO2 in the lungs over here. Here's the PO2 in the tissue, and you see at different pH levels, what you see is a shift to the right as the pH decreases, and the pH will be lower in actively respiring active tissue because they're producing CO2, which we know is acidic, okay? The last thing, you guys, is a thing called BPG, okay, biphosphoglycerate. It has multiple names. And what BPG does is it actually causes hemoglobin to release oxygen. It shifts the curve to the right. Now, it turns out that fetal hemoglobin doesn't really bind BPG at all, and that explains why the fetal hemoglobin is shifted to the left of hemoglobin A. So we have BPG, we have protons, we have CO2. Those are all things that cause hemoglobin to release its oxygen. BPG is derived from glycolysis. I won't sweat this too much. It's not the same as 1,3-biphosphoglycerate. It's actually converted by a mutase into BPG, so it's made in red cells. And the last slide, I believe, is that hemoglobin actually binds with CO2. It doesn't just carry oxygen, okay? What you have is a valine residue on the N-terminus that actually gets carbamylated, they call it, and you carry it over to the lungs, and when you get into the lungs, this spontaneously dissociates. This is a non-enzymatic reaction. About 20% of the carbon dioxide that you get rid of is attached to hemoglobin at one time, okay? I apologize, I thought this was the last slide was this one. This is a hemoglobinopathy. We're gonna see this today. This is really high yield. So you guys understand hemoglobin, you understand how it works, but there's a mutation where you take a glutamate and you convert it into valine, okay? The notation is E6V, as glutamate 6 goes to valine. And it turns out that when this is in a deoxy form, by making that hydrophobic valine, what happens is that there is a polymerization event that occurs. And when it polymerizes, it precipitates in the red cell, it distorts its membrane, and that's actually causal for sickle cell anemia. So sickle cell anemia is a mutation from glutamate to valine. You should definitely memorize that. By going from glutamate to valine, you've changed the charge, you're more hydrophobic, and it turns out it polymerizes and it becomes insoluble. That will be the last slide that we got. So at this point,